So you continue, and
Okay, sir.
if you can, you continue for 2 hours. Otherwise, you let me know. I will take the second hour.
I'll take till 12 o'clock, sir.
Okay, continue.
Okay, sir. Thank you, sir. Yeah. Good morning, all of you. Can you hear me?
Morning, ma'am.
Yes, ma'am. Good morning,
Good
ma'am.
morning, ma'am.
Good morning.
Morning, ma'am.
Good morning,
So,
ma'am.
I
Good
hope
morning,
what
ma'am.
we have taken yesterday, you all remember what we have
Good
taken
morning, ma'am.
yesterday. Good morning.
Yes, ma'am.
All of you are there
Yes, ma'am.
Yes, ma'am.
yesterday? All
Yes,
of you are
ma'am.
there? Okay. You all remember what I was taking yesterday?
Yes, ma'am.
We were discussing about the difference between a prokaryotic cell and a eukaryotic cell. Do you all remember what we were discussing?
Yes, ma'am.
One second. I'll just open the PPT for yesterday's. Can you see the screen now? All of you?
Yes, ma'am.
Yes, ma'am.
Okay. So we were discussing the difference between prokaryotic cell and eukaryotic cell yesterday. I hope you all remember whatever we have done. Can anybody tell me? I told you we'll revise tomorrow again. Can any one of you tell
Yes,
me?
ma'am.
Yeah, please tell who is going to tell me.
Cell size of prokaryotic cells will be from 1 to 10 micrometers, and of
Yeah.
eukaryotic cell is 100 micrometers.
Yes.
Nucleolus is absent in prokaryotic cell, whereas present in eukaryotic cell.
Yes. Everybody is listening, I hope. What else?
Ma'am,
What else? Who
ma'am,
else will tell?
Nucleus is not bonded by nuclear membrane in prokaryotic cell.
Hmm. And?
Nucleus is bonded by nuclear membrane in eukaryotic cell.
Yes. What else? Anybody else can tell me? I think
In
Muzaffer is telling, no?
ribosomes, ma'am. In prokaryotes, 70s ribosomes. In eukaryotes, 80s ribosomes. And
Yes.
internal membranes in prokaryotes are simple, whereas in eukaryotes they are complex.
Okay. What else? Who else will tell me? Akhila is there.
Membrane-bound cell organelles are absent in prokaryotes, whereas present in eukaryotes.
Okay. Who else will tell?
Golgi
Zygote
apparatus present in
cell.
eukaryote, absent in prokaryotes.
Yeah, that is Muzammil, no?
Harsha, ma'am.
Harsha, okay. Muzammil was
In
also telling like
prokaryotes,
in the
zygote
beginning.
is partially diploid,
No,
ma'am. In
no
eukaryotes,
mitosis.
diploid, ma'am.
Okay. Who else is going to tell me? Akhila, are you there?
No mitotic division in prokaryotes,
Hmm.
whereas mitosis will occur in eukaryotes.
Hmm.
And sexual reproduction in prokaryotes will be by fragmentary process.
Hmm.
And in eukaryotes, meiosis occur as a regular process.
Okay. What else?
Respiration occurs through cell membrane in prokaryotes, whereas takes mitochondria in eukaryotes.
Who else will tell me? Next points. Pawan, can you tell me?
DNA arrangement in prokaryotic cells are circular. In the case of eukaryotic cells, they are linear.
Okay. And Anjali, can you tell me? All of you tell one point, okay? Anjali, can you tell me? Anjali, you were there yesterday's class?
Yes, ma'am. There is network problem. Your voice is tracking.
Others are able to hear me?
Yes, ma'am.
Yes, ma'am.
Yeah, ma'am.
Okay. Anjali, you were there
Yes, ma'am.
yesterday?
Yes, ma'am.
Okay. Can you tell me any one point? One point at least. Nothing? No, you don't remember anything?
No, ma'am.
Okay. I told you all to note down the points if you cannot remember because there are many points. I told you all yesterday also to write down a few points because it's not possible for you all to remember so many points. And after the class, then you tend to forget. Naveen, can you tell me anything? Naveen is there. Naveen, you were there yesterday also. If you don't answer, I consider you as absent. At least answer. Naveen, you're not there. You can just not switch on and go. You should be there in the class. If you don't answer, you're considered absent. Naveen. So you're not there. You should answer. You know or you don't know, you should answer. If you're not answering means you're not there. You've just kept it open. Okay. Amarnath, are you there? Amarnath,
Yes, ma'am.
yeah. Can you tell me any one point? This person,
Pseudopodia,
huh?
ma'am. Pseudopodia is absent in prokaryotic cell, present in some eukaryotic cell, ma'am.
Okay. You were there yesterday,
DNA,
no?
DNA base ratio as mole of eucan and plus cytosine 28 to 73 in prokaryotic cell, about 40% in eukaryotic cells.
Shiv Prasad, can you tell anything?
Appendages, ma'am. In prokaryotes, pili and flagella. In
Hmm.
eukaryotes, flagella with whip-like structure.
Hmm. Yeah. Anything we have left out on the points we discussed yesterday? Nobody told me endoplasmic reticulum and
Prokaryotic lacks sterols,
Chloroplast, ma'am.
whereas eukaryotes have
Chloroplast,
sterols.
mitochondria.
yeah, yes, yes. You forgot endoplasmic reticulum, Golgi body, and what
Cell
else?
size in eukaryotes is large, ma'am, 10 to 100 micrometers. In prokaryotes, less than 5 micrometers.
Yeah, that was I think first told by Muzammil. Cell size, cell size was told first by Musammil, I guess. Okay. Uh, Anything else we missed on? You all
Histones
have
are
covered
absent
all
in
the points.
prokaryotes, whereas
And endo and
Who else is there in
exocytosis
the
is
class?
absent in prokaryotes, whereas present in
Pranay?
eukaryotes.
yes, ma'am.
I didn't hear that. What did you say,
Endo and
Muzammil?
exocytosis is absent in prokaryotes, whereas present in pro- eukaryotes.
Hmm. Okay. Okay. So, Pranay is there in the
Absent
class?
yesterday, ma'am.
Yeah. Pranay, you were there yesterday? You were not there yesterday? Pranay was
No,
present
ma'am, I'm
yesterday?
absent yesterday.
Absent. Why absent?
I went to marriage, ma'am.
Don't miss classes because all these classes, your first, we are taking first unit and second unit. Sir is taking first unit and I am taking the second unit. And all important topics. Most of you you have studied in 12th, but you don't remember. Most of you don't remember whatever you all have studied. And we are doing a recap. And also, this is much in depth. These topics, when you're doing at your UG level, they are much in detail. Okay, so don't miss classes. I'm telling all of you, and you just switch on the phone and go. That will also not do. Concentrate and listen whenever the class is taken. Okay, and I think this slide we have done yesterday. This slide we have to do. Plasma membrane. We have
Madam,
not
9 plus 2 array
discussed
is
about
found
the.
in eukaryotes, whereas it's absent in prokaryotes.
We have discussed this. Yes, we have done this. So, and I think this one we have to do today. In prokaryotes, it's gliding motility, gas vesicle mediated movement. And in eukaryotes, it is cytoplasmic streaming, amoeboid movement, and gliding motility. I think I've done this yesterday.
Yes, ma'am, we
Appendages,
have done.
villi, and flagella. Yeah. And in eukaryotes, flagella with whip-like motion. It is repeating cytoplasmic streaming absent and present in eukaryotes. Okay, locomotive organelles. It's a simple fibril over here. Locomotive is for motion, movement, and 9+2 configuration. So it can be more than one. Pseudopodia absent and pseudopodia present in eukaryotes. DNA ratio is 28 to 73, and here about 40%. And all this we have done. Mitochondria, chloroplast, Golgi, and all this is absent in prokaryotes, and all this present in eukaryotes. Okay, so there are, I think, more than 20 points. Did anybody write down? Anybody knows how many points we have covered? How many are there? 10, 20, more than 20, 25 points are there. So you need to remember all these points. It's not when anybody is asking you preference between prokaryotic and eukaryotic cell, you can just not say 1 or 2 points like you told me in the first class. There are almost 25 points of differentiation and you need to remember all these points. Is it clear, all of you? Is
Yes,
it
ma'am.
clear? Remember
Yes,
all
ma'am.
these points which you were telling. Okay. Next comes difference between gram positive cell walls. We will be doing between the gram positive cell wall and gram negative cell wall. We have discussed the structure of the cell wall in the previous class. I have showed you the structure. You'll remember the structure, no? I've showed you the slides of structure of gram positive cell wall and gram negative cell wall. Yes or no?
Yes, ma'am.
Yes, ma'am.
Yes. Yesterday also I showed you the cell wall pictures, gram positive and gram negative. Okay. So these are the points of difference between the two cell walls, gram positive cell wall and gram negative cell wall. Okay. So here cell wall is distinctly thick. I told you in gram positive the cell wall is distinctly thick, and here whereas it is comparatively thin in gram negative. Okay. And in gram positive, note down or you'll tend to forget. Note down the points or you'll tend to forget. Cell wall is one layered, and here the cell wall is two layered. But difference is the peptidoglycan in gram positive is 90%. I showed you all the images, but the peptidoglycan is a big thick layer. The NAG and NAM bonds linked with the oligopeptide bridges. I've shown you the image yesterday. What is NAG and what is NAM? Pranay, can you tell me what is NAG and what is NAM? Anjali, can you tell me what is NAG and NAM? Anjali,
Yes, ma'am.
can you tell me what is NAG and NAM? Yesterday I took it. You were there yesterday, no?
Yes, ma'am.
I took it in the previous class also. Yesterday I repeated again. What is NAG and NAM? Shiv Prasad,
Yes,
can you
ma'am.
tell me?
N-acetyl glucosamine and N-acetyl muramic acid.
Yeah, N-acetyl glucosamine and N-acetyl muramic acid. Anjali, what happened? I've repeated so many times. Repeated that in two classes, yesterday's and previous class also. Concentrate, that's what I'm telling you. Concentrate whenever the class is there so that you don't forget. Listen carefully.
Okay, ma'am.
So here it is a peptidoglycan layer which is comprised of 90% of the cell wall, and here the peptidoglycan is up to 10% of the cell wall. Cell wall is composed of higher amino sugar contents, and here less amount of sugars. Teichoic acid may or may not be present in gram positive, and teichoic acid is present absent in gram negative. Okay, it's completely absent in gram negative. Gives negative charge to the cell wall. Okay, here the lipopolysaccharides give the negative charge to the cell wall. Okay, that day who was telling me about this negative charge, I think it was Deepak, no? When we were talking about staining, someone was telling me about the negative charge of the cell wall. Okay,
Yes, ma'am.
so outer membrane is absent in case of gram positive, and outer membrane is present in case of gram negative. Periplasmic space is absent. In the figure I showed you yesterday, the structure, periplasmic space was not there in case of gram positive, whereas it was present in case of gram negative. Hafsa, समझ में आ रहा है? Hafsa,
Yes, ma'am.
you understanding, no?
Little, ma'am.
Little.
Yeah, ma'am, because everything is new, and the pronunciation, and I can't mention more because everything is new.
The subject is
I
new or the
no, ma'am. All subjects is new for me.
all subjects are new?
Yeah, ma'am, because I there is no intermediate in my country.
Okay.
But I will do my best, and
Yeah, if your doubts, you can ask me. Okay, you can contact me personally, and whatever you don't understand, you can ask me again.
okay, ma'am. Thank you.
Because I'm going with I'm asking because whatever they've done in 12th for these people, they've done a bit in 12th, so they have an idea of everything, and
Yeah,
then I'm proceeding. But for you, I don't know. I will you whatever you have problem, you can contact me personally. Okay.
okay, ma'am. Thank you.
Okay. You understand when I talking? No, you get it? No.
Yes, ma'am. But after classes, all classes, I have to translate it because everything I don't know in like in Arabic.
English, you can do it, no?
No, not more, ma'am. Because, ma'am, there is difference between my studies and the Indian studies. In India, all people has notes because they study intermediate. But me, no, there is no this subject in my country. But
Okay.
okay, but I will do after. I will do my best after classes.
Okay. We'll you get back to me. Okay.
Okay, ma'am.
The class. Okay. Hmm? Whenever you feel that I need to repeat, you tell me, I'll repeat it. Okay.
Thank you, ma'am.
Points and periplasmic space is present in gram negative. Okay. Little amount of lipids, that is 1 to 4%, and lipids form major component, that is 11 to 22%. Okay. That is in case of gram negative. Okay. And then few amino acids are present in cell wall, whereas large amount of amino acids are present in case of gram negative cell wall. Cell wall can be digested by lysoenzyme. Lysoenzymes react only after pre-treatment. Okay, and they are penicillin sensitive. Gram positives are penicillin sensitive, and here they are penicillin insensitive. Okay, magnesium ribonucleate are present in case of gram positive, whereas absent in case of gram negative. Inhibited by high concentration of crystal violet, unaffected by crystal violet. Okay, I told you, I explained you the crystal violet, what happens when we do staining. I told you all this crystal violet penetrates in the cell wall, and because of the high amount of peptidoglycan, it remains in the cell wall and doesn't move out after washing, also because of the peptidoglycan layer, thick peptidoglycan layer. Okay, and here crystal violet gets washed off. Okay, I explained you all this concept. Not dissolved by 1% of KOH, and gram negatives get dissolved by 1% KOH. KOH, what is KOH? What is KOH?
Potassium hydroxide.
Potassium hydroxide. Potassium
Yeah.
hydroxide.
Yes, you all should know this, at least this, you know. Then exotoxins are produced, endotoxins, and in this case, gram negative endotoxins are produced. They are gram positives are spore producing rods and cocci. Okay, and these are non-spore forming. Yesterday we discussed about the spore. Hmm? We discussed about the spore. I showed you all the diagram for spores and how do we do staining for spores. I told you all, right? You'll remember.
Yes, ma'am.
You'll remember I told you all spore staining and all yesterday. So this is what gram positives are those which produce spores, and not in case of gram negative. Basal body of flagella consists of two rings. Okay. Basal body of flagella consists of four rings over here. I'll discuss in detail the flagella structure and all with you all. Do you all remember flagella structure? Anybody?
Yes, ma'am. A little bit.
Yeah. Okay, so the flagella consists, the flagella basal body consists of rings. Okay, in this case there are only two rings, and in this case there are four rings. Okay, this is clear difference between gram positive and gram negative cell wall.
Yeah, ma'am.
Did you all note down the points of
Yes, ma'am.
difference?
Yes, ma'am.
I hope you all taken the points of difference. Then don't forget. Okay, this won't be repeated again. This won't be repeated. Shall I close it? Shall I close it?
Okay, ma'am.
I hope all of you have taken this.
Yes,
I'll
ma'am.
go to the next PPT if you all remember this. Shiv Prasad, Pranay,
Yes, ma'am.
Anjali.
Yes, ma'am.
I hope all of you have taken this. This won't be repeated
Eh,
differentiation because I've taken two classes, difference between positive and negative. I hope you all remember this. And we have also discussed the difference between prokaryotic cell and eukaryotic cell. This is a major question which you get differentiate between gram positive prokaryotic cell and eukaryotic cell. You have to do it with structure. You have to draw the structure of prokaryotic cell, you have to draw the structure of eukaryotic cell, and then put the points of difference between prokaryotic and eukaryotic cell. Okay? Did you all go through the syllabus? Syllabus देखा?
Yes, ma'am.
Shall I close this PPT? You all have taken the important points?
Yes, ma'am.
Okay, ma'am.
Okay. Did you all go through the syllabus? No? I have posted a syllabus in the group.
Yes, ma'am.
You have seen the syllabus, I hope.
Yes, ma'am.
You all have not seen the syllabus. I'm taking the second unit. देखा है second unit में portion क्या क्या है? I'm asking you all. Have you all seen second unit?
Yes, ma'am.
What all portion is there? So I am taking the second unit and sir is taking the first unit as I told you all. How many of you don't have the syllabus?
I don't have, ma'am.
Who is that?
Harish, ma'am. I'm new to your class.
Harish or
Ma'am,
Harsha?
I too don't have.
Tell me your names.
Harish, ma'am.
Harsha. Harish.
Harish. Yes, ma'am.
Harish. Harsha is
Ma'am,
different. Harish is different.
Yes, ma'am.
Who else? Tell
Ma'am,
me your
Me
name.
also,
I
ma'am.
don't have,
Harsha.
madam. Pavan Kumar, madam.
Okay. So you all don't have the syllabus.
Yes,
Navyashree,
ma'am.
ma'am.
Navish, you all don't have.
Yes, ma'am.
Okay.
K. Navyashree.
So many of you don't have the syllabus with you all. It was
Yes, ma'am.
posted in. It was posted in the group. You all were not there in the group then.
I have joined newly, ma'am.
Okay.
I'm also, ma'am.
Tell me names.
Harish, ma'am.
Harish. Okay. Okay. Because I yesterday also, day before also, I asked you all did not tell me.
Yesterday I
I
did
asked
not.
if you all have the syllabus. You should have
Ma'am,
told me,
don't.
no?
No.
Ma'am, I have posted in the group, ma'am.
When? When did you
Now,
post in the
ma'am.
group? Now.
No, ma'am.
We have, ma'am.
Okay.
So we have posted.
Okay. That's what. If you all don't have, you should tell me. I was asking in the previous class also, no, if you all have or you all don't have. You should tell me if you all don't have. Okay. So we'll start with another PPT for classification. Okay. That's what, because you have cell structure, then you have differentiate between the cells prokaryotic and eukaryotic. Then you have difference between gram positive and gram negative. Okay. So we have finished with difference. We have finished with all this. We'll be doing classification. I'll start with the next PPT. Can you all see the screen?
Yes,
Yes,
ma'am.
ma'am.
Yes,
Yes,
ma'am.
ma'am.
Okay. Okay. So as discussed, now in your syllabus, you have to study classification by Calhoun, classification by Whitaker, classification by Haeckel. Okay, have you all heard these scientists?
Yes,
You all
Yes,
have
ma'am.
heard
ma'am.
these
Yes, ma'am.
scientists?
Yes,
Yes,
ma'am.
ma'am. We
You
have
all have
in
heard?
intermediate, ma'am.
Yeah, you have an intermediate. So we have all these three classifications. Did you do in detail? You have studied the classification by Whitaker, Haeckel, and Calhoun in detail.
Whitaker's
Yes, ma'am.
classification was studied in detail.
Okay. Then Haeckel and Calhoun.
Heckle and Carlos not in detail, ma'am.
Haeckel and Calhoun you all have not done. Okay. So Haeckel was a German scientist. Okay, he divided the he classified the prokaryotes into three kingdom group. Three kingdom group. Then there was Calvus, and then there was Whitaker who did five kingdom group. So we have to study all these. Now, first I'll explain you with pictures, and then we'll do the tabular ones. Okay. So here the five kingdom group. that is Monera, protists, plants, fungi, and animals. Then that is the domain. They have three domains, three new domains were created. Okay. In that they include what? They include the prokaryotes and the eukaryotes. Okay. See, these are the images. These are the images. Protista, archaebacteria, bacteria, eukaryotes, and prokaryotes. We need to study everything in detail. Here you have to study the characteristics also. You have to study the characteristics of archaebacteria, the characteristics of protista. All this is there in your syllabus. Okay. See, this is bacteria, archaebacteria. What is archaebacteria? Anybody knows? What are archaebacterias?
Yes, ma'am.
It
Which
can
can
survive
survive
in
in
extreme conditions.
extreme habitats.
Yeah, can survive in extreme conditions. Then you can see they can survive in volcanoes, they can survive in underseas where the salt concentration is high. Okay, they can survive in hot water streams. So archaebacteria are those which survive in tough conditions. That is where the temperature is high. Also, they can survive. They can survive when the acidic, and then when this acidity is more, like acidic conditions are more. That is in the depth of the sea, ocean beds. Okay, so it can survive over there also, and in hot water springs also they can survive. So that is the archaebacteria group. So that is the reason they survive for many decades. Then this is the kingdom protist, kingdom plant, kingdom fungi, and kingdom animal. This is the five kingdom group. Then prokaryotes. When we are studying prokaryotes, we are studying archaebacteria, eubacteria, and bacteria. Okay. Bacteria. When we are talking about bacteria, they live in all ecosystems. Okay. We know we have discussed about this. Coming to UG level, you know that bacteria survive in all ecosystems and everywhere. Okay. So they're in soil, plants, and they're in depth of oceans, extreme cold conditions, salt conditions. Okay, on the dead, everywhere. So we are studying the different categories of bacteria where, okay, depending upon their ecosystem. Now these are the shapes what I discussed yesterday with you. This bacillus, I was discussing the shapes with you yesterday. I couldn't show you these images. So today it's here. Okay, Tocobacilli, then this is Clostridium. This produces spores. Okay, we discussed about spores yesterday. Spores we were discussing yesterday. You remember?
Yes,
Yes,
ma'am.
ma'am.
You'll remember? Spores. We discussed endospores yesterday. We discussed the staining produce the spores. Okay. So when we do spore staining, you can see the spores inside. This is E. coli. Well, I've heard about E. coli.
Yes, ma'am.
Yes, ma'am.
E. coli. E. coli, you all have heard about. Which other bacteria names do you know? Can anybody tell me names of any bacteria if you all have heard?
Astobacter.
Astobacter, Aspergillus,
Astobacter.
Nostoc, Anabina,
I'm asking bacterial name. Who said this? Tell me your name. Who said this?
Muzammil,
Okay. Bacteria names. E. coli.
Lactobacillus bacteria.
Lactobacillus. Who said that?
Harish, ma'am.
Harish. Okay. Harish, you're coming today or yesterday you were there?
Today only, ma'am.
Today only. Why you were added in the group long back?
Yesterday only I get link, ma'am. I'm new to your class.
Okay. You were added in the group when?
29th, ma'am. 29th December.
Yesterday.
Yes, ma'am.
Okay, whatever. So you have missed the classes taken by sir, yeah, HOD sir.
Yes, ma'am.
Yesterday morning, did you attend his class?
Morning I attended, ma'am.
9 o'clock class, did you attend?
Yes, ma'am.
Yeah, because he is teaching history of microbiology and he is taking lots in depth. So the important classes you all should not miss.
Louis Patcher
Okay.
and Robert Koch.
Yeah, he took
Yesterday.
that. He took Edwards and also he took, I guess.
Yes, ma'am.
Yeah. Okay. So lactobacillus, you told me. Any other bacteria name? Can you tell me?
Cyanobacteria, ma'am.
Cyanobacteria. Who said that?
Arachnobacteria.
Gautam, ma'am.
Gotham. Okay. Any other names?
Bacillus anthracis.
Bacillus. And it's good to hear names of bacteria from you all. Was it introduced to you all in 12?
Yes, ma'am.
Yes, ma'am.
Okay.
Corynebacterium, ma'am.
Corynebacteria. Yes,
Mycobacterium.
good. Who said that?
Gautam, ma'am.
Good. Good. Who said that? Who is saying? You can tell your names, no? Okay. Girls, you also have studied, I guess. None of the girls know. Penicillin, very common one. Penicillin notatum.
Bacillus
It
anthracis
comes as a
bacteria.
fungus group. Yeah. See, here are the names. Salmonella is one. Streptococcus is one. Staphylococcus is one. Okay, they are in front of you. E. coli is in short, Escherichia coli in short is told as E. coli. You know that?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
Yeah. So this is you know how to write the species and the genus.
Yes, ma'am.
You know
Yes,
how
ma'am.
to species and
Yes,
the
ma'am.
genus.
Yes,
So
ma'am.
this is when you say E. coli, E is capital, then you put a dot and write coli. That is the okay. E. coli when you
Yes, ma'am.
say, and then when you say salmonella, you have many species in salmonella. It's not only salmonella. You know that?
Yes, ma'am.
Yes, ma'am.
Salmonella, they have many species in salmonella. So streptococcus also, they have many species in streptococcus. Staphylococcus also, there are n number of species in staphylococcus also. Okay. So what you do, you write capital S, then you put a dot, and then you write the species name. Okay. So S will stand for salmonella. You write capital S dot the species. That is how we write the species name. Streptococcus will have many species. Staphylococcus will have many species. Okay, so see this. I told you E. coli are negative. E. coli are gram negative, and when you stain them, also they become pinkish red. Hmm? So it is almost this color when you stain it. You can see small rods over small cocobacilli over here like this. You can see. Hmm? When you stain it, it will be reddish pink. Then salmonella, okay. Streptococcus, yesterday I told you which is in chains, which is in bunches. When I explained you the structures of bacteria, I told you to remember which will be in chain, which will be in bunches. Which is in chains and which is in bunches.
Streptococcus in chain and Staphylococcus in bunches.
Streptococcus is always in chains, okay, and Staphylococcus is in bunches, okay. So you have to remember that Streptococcus, when you see under the microscope, you will see it in chains. It will be cocci. It will be cocci in chains. You can see many chains over here. Hmm? When you see Staphylococcus, it will be in bunches. And once you see staphylococci, it will be present cocci. The shape will be cocci, but it will be present. Clear, all of you? Hafsa, is it okay?
Yes, ma'am. Yes, ma'am.
Understanding the shapes with figures I've shown you here. See, these are different bacteria names. You're there in the class, I hope.
Yes, ma'am.
Okay, so I'll go to the next slide. Hmm? If this slide is clear, I'll go to the next slide. Okay, all of you? Anjali,
Okay, ma'am.
Akhila. Okay,
Yes, ma'am.
so
Yes, ma'am.
this, these are the cells, prokaryotic cell structure we have done in detail in the previous PPT, yeah, and eukaryotic cell structure also we have done in detail in the PPT in the last class. Okay, and here comes the size 1 to 10, 1/10 size of eukaryotic cell. Okay, when you're studying the prokaryotic cell, it will be 1/10 the size of eukaryotic cell. Okay, internal structures we discussed just now. No internal compartments, no membrane bound organelles, and only ribosomes. You can see the structure over here. It has no internal compartments. Like in eukaryotes, you have internal compartments, you have membrane bound organelles over here in eukaryotic cell. Okay, but it's not that case in prokaryotic cell. It's only the ribosomes which are present in the cytoplasm. Hmm? Circular chromosome and naked DNA. The DNA is not enveloped into a nucleus. We discussed this. The DNA over here is free floating in the cytoplasm. It is not enclosed inside the nucleus, whereas here the DNA will be inside the nucleus. Okay, genetic material. Then coming to the DNA, double helix structure. See the double helix structure. What was it here? Circular, but what was this? Single stranded circular. Hmm? Whereas here it is not like that. It is a double helix in eukaryotes. Can you see this? Can you all see this diagram?
Yes, ma'am.
Yes, ma'am.
Understand the
Yes,
strand
ma'am.
DNA structure? Have you all, do you know DNA structure?
Yes, ma'am. Double
Yes,
helix
ma'am.
structure.
Yes.
Double helix, and what is it? The bonds bonds are
Sugar phosphate bonds, ma'am.
which are the bonds? What are the DNA bonds?
Phosphodiester bond.
ATGC. ATGC.
Adenine, guanine, cytosine, thymine.
Yeah.
Adenine, thymine, guanine, and
AT,
cytosine.
yeah.
Hydrogen bonds, ma'am.
ATGC. ATGC, and backbone is
Hydrogen
sugar
bonds.
phosphate bonds. Backbone of the DNA is the sugar phosphate
Sugar and
bonds.
phosphate.
ATGC, what is A, what is T? Adenine,
Adenine,
Adenine, thymine.
thymine,
Adenine,
Thymine.
Adenine, thymine,
guanine, and cytosine.
guanine,
yeah.
cytosine.
Yeah, that is in case of DNA. In case of RNA, what is replaced?
Uracil.
Uracil.
Replaced by
Thymine is replaced by uracil.
thymine
right?
replaced by uracil.
One girl answering for the first time. Who is that? Who is answering? Tell me your name. None of the girls are answering. Only one I heard just now today. Generally, it's the boys only answering in the class. Okay. So RNA, in RNA, thymine is replaced by
Uracil.
uracil. Okay. Yeah. So you will be doing this structure in detail. You have a DNA structure in detail in your next second year. Okay. But now, for now, it is. Can you see the strand? In prokaryotes, you have a circular single-stranded one. Hmm? Can you see this strand,
Yes,
all
ma'am.
of you?
Yes,
Yes, ma'am.
ma'am.
So
Ma'am.
this is enlarged figure for those who have not understood or for those who don't understand. The this is a picture. Hafsa, I think this should make it clear for you. This is the DNA double-stranded structure over here. Okay, and here this is the enlarged view of it. This is this strand. This strand, you can see the enlarged view of this. Okay. So when you are saying it is a DNA in the nucleus, it is DNA in the nucleus. It is like this. Okay. Clear, Hafsa?
Yes, ma'am.
Yeah. And here what happens? This chromosome. So in eukaryotes, you have chromosomes. You have chromosomes in eukaryotes. And on
Yes.
the chromosomes, you have the helical structure over here. The DNA is wound circularly over the Can you all see this? Eukaryotes, you have the chromosome and the DNA is wound, circularly wound over the nucleosome. This whole thing is called as the nucleosome. And the DNA is wound circular over this. Can you all see this diagram?
Yes, ma'am.
Yes, ma'am.
Yeah, I think this diagram should make it clear for you all. For some have the doubts where the DNA is actually present in the cell. This structure, where is, and these are the bonds. You can see these bonds, ATGC bonds. These are the ATGC bonds. Okay. And can you all see over here, nucleosome? This complete structure is called as nucleosome, and the DNA is wound, is present on this. It is circularly wound over this central histone and spacer histones. Okay, these are called as histones, and this is the central histone, and this is the spacer histone. Clear, all of you? You should
Yes,
not
ma'am.
have any doubts about this, where it is present, how it is present. Okay. In case of prokaryotes, it is just the strand which is present in the inside the cell. In case of eukaryotes, it is on the chromosomes that the DNA is wound circularly over the histones. Hmm? I'll go to the next slide. I hope it's clear.
Yes, ma'am.
Yes, ma'am.
Okay. Then coming to different kinds of bacteria. Coming to cyanobacteria. Cyanobacteria is the photosynthetic bacteria. Cyanobacteria is the photosynthetic bacteria. So internal membranes produces like chloroplast. Okay, this will have chloroplast inside. Internal membranes like chloroplast are present in case, and this is a photosynthetic bacteria. Cyanobacteria is the photosynthetic bacteria. And aerobic bacteria, I told you what is the difference between aerobic and anaerobic yesterday.
Yes, ma'am.
Yeah, what is the difference between aerobic bacteria and anaerobic bacteria?
Aerobic bacteria
Aerobic
requires oxygen for its
oxygen.
growth.
Hmm. For survival, they require oxygen. And anaerobic, what do they do?
Anaerobic bacteria can
They
survive without oxygen.
Yeah, anaerobic bacteria can survive in the absence of
Can
oxygen.
survive without oxygen.
Yeah, anaerobic can survive in the absence of oxygen.
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 So when we grow in the lab, when we are growing in the lab, how do we do it? We grow aerobic and anaerobic. No, we'll be growing. When you come to lab, we'll be growing bacteria in our labs. All these bacteria, whatever you have studied, I have shown you in the slide, Staphylococcus, Streptococcus, everything you'll be growing in the lab. So, for aerobic, what will you do? And for aerobic, no idea. They are placed inside a closed jar. Anaerobic bacteria are placed inside a closed jar with completely sealed and no oxygen for them. Okay, it is seen to it that oxygen is doesn't enter the jars. Hmm? So it is placed in a closed jar, anaerobic bacteria, for them to grow. We'll do all this when you come to lab. You can see this mitochondria. These are the what? Mitochondria are the
Powerhouse of the cell.
The
yesterday you all
cells.
powerhouse of the cell. Okay, they are responsible for the respiration in an aerobic bacteria. Okay, I mean this is not for in the case of this is in eukaryotes. Okay, not in case of prokaryotes. This is in case of eukaryotes. Prokaryotes do not have mitochondria. Here the cell walls, cell membranes act for respiration. Yesterday we discussed that. Hmm? And in case of now, this is gram positive and gram negative. Once again, a repetition. Okay, so this is a part of the cell, how it looks, a part of the cell wall, a part of the cell wall after staining. This is how I told you, crystal violet, when it takes a stain, this is how it looks. And gram negative, a part of the cell wall, when it stains, it looks like this. Harsha, it is a repetition for you. And for those who have not heard this before, this is how it is. And this is the cell wall. Okay. The peptidoglycan is polysaccharides plus amino acids. What are the amino acids? What are the amino acids? Just now you all told me. You just now told me what are the two amino acids? Peptidoglycan layer, two
NAM
amino
and
acids.
NAG.
Yeah. Who said that?
NAG.
That is what I'm asking you all. N-acetylmuramic acid and
N-acetyl-muramic
N-acetylglutamic
acid.
acid. Yeah. So these are the two amino acid chains. They are bound by oligopeptide bonds. We have discussed this yesterday also. And lipopolysaccharides are lipids plus polysaccharides. Okay. And here in gram positive, the peptidoglycan layer is thick. We have a thick peptidoglycan layer. See, this is the thick peptidoglycan layer over here. And then the plasma membrane. And these are the proteins which are embedded in the plasma membrane. So this is a complete structure of the peptidoglycan layer. Hmm? Over here, it is the outer membrane is of lipopolysaccharides. This is the outer membrane of lipopolysaccharides, whereas the peptidoglycan layer is very thin. You can see this. It's a single thin layered peptidoglycan layer, and this complete is the this makes up the cell wall. Okay, the outer membrane and the peptidoglycan makes up the cell wall. Outer membrane here is composed of lipopolysaccharides. Then comes your plasma membrane. This is the fluid mosaic structure we have discussed in the previous class. Remember, all of you?
Yes, ma'am.
I hope you all
Yes, ma'am.
remember. Okay. And these are the different bacterias. This is photoautotrophs, chemoautotrophs, and heterotrophs. You know the
Yes, ma'am.
difference, no? Photoautotrophs, chemoautotrophs, and heterotrophs. I hope you all know the difference. Photoautotrophs,
Yes, ma'am.
Yes,
chemo,
Yes,
ma'am.
ma'am.
They
Yes,
chemo,
ma'am.
derive energy
photo,
from sunlight.
yeah, chemo
And
at the
carbon source from carbon dioxide.
yes, and these photo, the term is for photosynthetic bacteria. Okay, they take it from sunlight. Chemo, the term, they take the energy from oxidized inorganic
Oxygen.
compounds like nitrogen, sulfur, hydrogen, etc. Okay, and then the heterotroph, which live on plants and animal matter, and also on decomposers and pathogens. Okay, getting it, all of you? So these are the
Yes, ma'am.
pictures
Yes,
show
ma'am.
you the
Yes, ma'am.
photosynthetic bacteria, the chemoautotrophs, which take the energy from oxidized inorganic compounds, and these are other bacteria which survive on decomposing and plant and animal matter. Okay, they come under heterotrophs. Clear?
Heterotrophs.
Parasites, saprophytes.
Yes, yes. Can you tell me your name? How will I know who is talking? Hmm? I'm asking whoever is answering, please tell the name so that I know who is answering. Okay, genetic variation in bacteria. You'll know this mutations, genetic
Yes, ma'am.
recombination,
Yes, ma'am.
conjugation.
Yes,
You
Ma'am,
also
ma'am.
we
know
have
this. See,
studied in
in this
intermediate.
bacteria, you can see over here the two with the pili. There's a transfer of genetic material. You can see the two bacteria as an electron microscope view. Only then you can see the pili. Okay, this is the pili, and the genetic material gets transferred from this one bacteria to the another.
To
Okay.
that bacteria.
Yes, and this is the conjugation. This is the binding to bacterial cells. We'll do all this in detail in your next chapters whenever we go through the conjugations. This is a big thing. you'll have next year. Okay? And this is not cause any harm to you all. Okay? Bacteria, I hope you know that. Yes or no? Are all the bacteria are harmful to us? It's not like that, that all are harmful to us. Yes or no?
Yes, ma'am.
Yes,
Yes,
ma'am.
Yes,
ma'am.
ma'am.
There are
There are
some
a
friendly
few.
bacteria.
There are a few which are good for us also. Can
They
you
are
tell me?
friends and foes for us.
Yeah. Can you tell me any name of those bacteria which are not harmful to us and which are good for our health?
Lactobacillus.
Yeah, very good. Who said
But
that? Who said that?
Sri Akshara, ma'am.
Shri Akshara. Good. Okay. Instantly telling an answer is good. Telling after some time is also good. But if you stay instantly means you are confident that it is correct. Hmm? Okay. Any other bacteria? Any other bacteria can you mention?
rhizobium bacteria, ma'am.
Rhizobium. Yeah,
Penicillin.
it is not harmful.
E.
Good.
coli, ma'am.
Penicillin.
E.
Penicillin,
coli
ma'am.
is harmful to us. Penicillin, yeah, it doesn't come under, yeah, is fungi. And any other bacteria? Have you heard about probacterias, probiotic drinks? Have you heard
No,
about
ma'am.
this?
No, ma'am.
Yes, ma'am. Yakult.
Yeah, you have told the name of the brand. What is that?
Yakult, ma'am.
Yeah, it is a drink, probiotic drink. You'll have heard others.
No, ma'am.
You get the probiotic drinks in market. You get the probiotic drinks in market. You know that?
Yes, ma'am.
You should know that because nowadays probiotic drinks are so much in market and people consume it because consuming those bacteria is good for your health. Okay, it is good for your health and it increases your body resistance and mostly it helps in gastric problems. Okay, so it has more amount of protein also. These probiotic drinks have more amount of protein compared to other drinks. They have more amount of protein, so people consume it and it is much in demand nowadays. Probiotic drinks, other than the usual stuff, people buy the probiotic drinks so that it's good for your health. It increases the protein amount, it increases the bacterial content of your body, and you need those bacteria for the good health of your body. Okay, so it is not that all bacteria are harmful for us. There are few range of bacteria which are also good for your health, and they also some also make up the immune system strong. Okay, so those are a range of bacteria which you need. Your body needs them. Hmm? And then there is a wide range of bacteria which cause animal diseases, plant diseases. Okay, so that we are aware of. The number of diseases what we get throughout is all because of these bacteria present. Pseudo TB, okay, pneumonia, Lyme disease, typhoid. Typhoid, which bacteria?
Salmonella
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 type.
typhi.
typhi.
Yeah, who said that?
Salmonella
Salmonella
typhi.
typhi.
Yes, Salmonella typhi. Okay, Salmonella typhi is responsible for typhoid. Cholera.
Vibrio
Vibrio
cholera.
cholera.
Vibrio cholerae,
Bacteria
ma'am.
cholera.
Yeah, very good, very good. So you all are aware of all this. Vibrio cholerae, very good. And what else you all can tell me? Anthrax, you all told me Bacillus anthrax. And what else?
Bacillus anthracis.
Bacillus anthracis.
Anthracis.
Yeah. And what else? TB.
Mycobacterium tuberculosis.
Mycobacterium tuberculosis.
Very good. Who told that?
Mycobacterium tuber.
Harish, ma'am.
Yeah, good. Pneumonia.
Streptococcus pneumoniae.
Streptococcus pneumoniae,
Streptococcus
ma'am.
pneumoniae
Yeah,
and
very good.
Haemophilus influenzae.
Very good. Streptococcus pneumoniae. Hemophilus influenza. Yeah, it is for influenza. Very good. So you all have a good idea of all the bacterias. So
Yes,
you all are
ma'am.
good microbiologists, I guess.
Somewhat, ma'am.
Yeah.
Somewhat.
If you all know all this in your first year, first class, so you all have a good idea of all the bacteria and what they cause. So good. You all will be good microbiologists. Good. And how about leprosy? Can you tell me?
Mycobacterium leprae.
Yeah, good. Who said that?
Harish ma'am.
Okay, so Harish has a good idea of all the bacteria. Good. And anything else? You all have left off? Anything you can think? Anything more you can tell me? That's it, huh? That finishes your kingdom of bacteria and their diseases. But this is good. You all know so much at this. It's good. Good knowledge. Okay. So those were about the bacteria which cause diseases and which are harmful to us. This is about those which are beneficial for us. Okay. This is about those which are beneficial for us. So there are recycling of nutrients from dead to living. Okay, these are the decomposers. The decomposers, they cause the recycling of the nutrients from the dead to the living. Hmm? Which is the bacteria for recycling? You all remember? Which is the one?
Nitrobacter.
Nitrobacter.
Nitrobacter. That is for nitrogen fixation.
Rhizobium.
Rhizobium. What does rhizobium do?
Nitrogen fixation.
Nitrogen
Fixation of
fixation.
nitrogen.
Nitrogen fixation. Yes. That is, these are the root nodules. You have done this. These are the root nodules on which the rhizobium is present. A rhizobium bacteria is present. Azotobacter rhizobium. And what do? They do nitrogen fixation.
Fixation.
Yes. Then this is what it is, nitrogen fixation and all. Recycling of nutrients from dead to living. Who will tell me? These are the decomposers. No, they live on the dead and they consume it. They recycle it. So which are that group of organisms which live on the dead?
Chemoautotrophic bacteria.
No,
Chemosynthesis autotrophs.
no.
Saprophytes.
Saprophytes. Heteroautotrophs. Tell me the name. You're telling the groups. Tell me the name. Can you recollect the bacteria? You have done this. I know you have done this. Which decompose the dead. Forgot?
Purple's bacteria.
Okay, go through and tell me tomorrow. Okay. And then E. coli is the one which helps in digestion, digests cellulose for herbivores, cellulose enzyme, produces vitamins K and B12 for humans. Okay. But sometimes it becomes harmful also. E. coli is also responsible for gastric infections. Okay. When you have gastroenteritis or anything gastric infections, E. coli is the one which is responsible. Then produce foods and medicines from yogurt to insulin. Then comes the lactobacillus. Lactobacillus, the one which comes in picture over here for yogurt. Okay, that's it. Till here, did you all understand?
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes,
Ma'am.
ma'am.
But we discussed about different bacteria, their uses, how they are beneficial and how they are harmful to us. We have discussed the different categories of bacteria which are harmful to us, which are useful to us. And we have discussed the structure of bacteria, the different categories, prokaryotes, eukaryotes, photoautotrophs, chemoautotrophs, heterotrophs. Hmm? All this is clear to you. We have done the cell wall structure. I've repeated many times. Gram positive cell wall, gram negative cell wall. I don't think you all should forget now. Is there anybody who still has doubts with gram positive and gram negative cell wall? I've repeated this almost in 3 classes. Anybody? Is it clear to all of you? This
Yes,
is the
ma'am.
basic of everything what you will be studying in your 3 years. All these first semester topics are basics of whatever you'll be studying for your 3 years. Okay, so don't have doubts in this more. If you have any doubts, tell me now itself. If you think you will have not understood, if you want me to repeat anything. So, anything anybody wants to be repeated, tell me. Cell structure, DNA, did you all understand where it is present? Hmm? I told you
On
all prokaryotes
chromosomes.
and eukaryotes. Prokaryotes, it is the strand which is in the cytoplasm. The DNA strand
Inside
is
of
there.
the cell.
Hmm. And in eukaryotes, it is in the chromosomes, or
Chromosomes.
it is wound over the histone. It is wound over the histones, okay, with central histone and spacer histone, and the complete thing known as nucleosome. The complete thing with the DNA is called as nucleosome,
Nucleosome.
and here the DNA is wound around the histone, central histone and the spacer histone, and this complete thing is is over here, the chromosome. On the chromosome you have the DNA. Clear all of you?
Yes,
Yes,
ma'am.
Pranay,
ma'am.
Yes, ma'am.
Yes.
Yes,
Anti,
ma'am.
Yes,
I
ma'am.
hope it's clear to you all. These are the different cells. Then these are the different bacteria images. I've shown you these images so that you have a better idea of all the bacteria. We have been studying the bacteria. You'll be seeing this once you come to lab. We'll do staining. We'll do culturing of different bacteria. We'll be growing all this streptococcus, salmonella, staphylococcus. All this will be growing in the lab, and then you will see them under the microscope after staining. Okay. We'll be
Okay,
doing
ma'am.
gram staining for this. We'll be doing gram staining, and then you'll observe these bacteria. If they're negative and positive, you'll be observing positive. Which color you'll observe?
Crystal violet, ma'am.
Yeah.
Crystal
And negative, you observe which color?
Red or
pinkish red.
pink?
Yeah,
Pink or reddish, ma'am.
or reddish pink. Okay. So at least my students should know this gram positive and gram negative concept clearly. When you come to second year and finally, you should not say what is it and how it is done. This is the basics of all. Okay, these are the different bacteria, groups of archibacteria and all. So clear? This is clear? All this?
Yes,
I'll
ma'am.
close this
Yes,
PPT.
Yes,
ma'am.
ma'am.
I'll close this PPT.
Yes, ma'am.
Yeah. Okay. You will take a 5 minutes break and start the next PPT. Okay. It's been 1 hour. You all are
Okay,
listening to
ma'am.
the class. You all are listening from past 1 hour. So I'll give you a 5 minutes break, and then we'll start with the next PPT. Okay.
Okay, ma'am.
Yeah.
Okay, ma'am.
Yeah. We'll take a 5 minutes break. You all are hearing from since 1 hour. I'll give you a break.
Right.
After 5 minutes, we'll start. Are you all back? Shall we start?
Yes, ma'am.
Yes, ma'am.
Yeah, okay.
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
I'll share the screen. I don't think you're able to see the screen. You all can see the screen.
Yes, ma'am.
Yeah. So I told you we'll be doing the classification of our organisms. Okay. I showed you the pictures so that when you'll be doing the tabular form, you understand. Hmm? Because the tabular form is very bland. You need to understand this. Hmm? And you have done all this, you're saying. Hmm?
Yes, ma'am.
So it should be easy for you all. Hafsa has not done, I guess. Hmm? Hafsa, I don't think you have done this.
No, ma'am.
Yeah. Okay. So this is classification of organisms. We have to study about three scientists: Haeckel, Whittaker, and Calvus. Okay. Haeckel was a German scientist, and was an American mycologist, and Calvus was also an American microbiologist. Okay. These two were American, whereas Haeckel was a German scientist. So he has given us three kingdom classification of organisms. Okay, Haeckel has given us a three kingdom classification of organisms. Classification is all about learning and remembering, and it is important. It comes in your exams. Classification is a major question in your exams, which you cannot skip. You have to be prepared with Haeckel, Whittaker, and Clawboost classification because they can ask any one of these three. Hmm? Any one of these three is asked, or any two, or like they can ask it for short, they can ask it for long. Yeah, okay, so you have to be prepared with this. You cannot skip it at any cost. It is all about remembering the classification. So Haeckel gave us a three kingdom classification, this German scientist. According to him, he has divided them into first two groups. Okay, all organisms lacking tissue system, morphological. This was in 18, okay, in the 18th century that Haeckel told us about this. He was the first one. So this was in the 18th century he was born. So this is all organisms lacking tissue system, morphological complexity, and division of labor. Hmm? This he divides into all organisms having diverse tissue system with well divided division, okay, of labor and maximum morphological complexities. So under this he kept the plantae and the animal group and the plantae kingdom he's kept under this. Hmm? And whereas here he's kept the protista, which includes all algae, fungi, protozoa, bacteria. All this comes under the protista group. I hope you all understand this. Hmm? It is
Yes, ma'am. Yes, ma'am.
the star group, which
Yes,
includes
ma'am.
algae, fungi, protozoa, bacteria. So this is the mainly protozoa and bacteria. Okay. So and here it is plantae and animals. So this is the three kingdom group given proposed by Haeckel. Was the first one to do this. Then plantae, under plantae it is you have autotrophic, and here it is holophagic mode of nutrition. Autotrophic mode of nutrition. We have studied the different modes of nutrition in 12th. Hmm? We did it in now also chemoautotrophs, photoautotrophs, heterotrophs. So here they are autotrophic mode of nutrition, and that is the kingdom plantae. And here these are all holophagic mode of nutrition. So they depend on others for the mode of nutrition. And under plantae, he has classified them into bryophytes, pteridophytes, gymnosperms, and angiosperms. This is also done in 12. Yes.
Yes,
Yes,
ma'am.
ma'am.
You study bryophytes, you study pteridophytes, you have done gymnosperms, angiosperms, everything. So this is the plantae group. If you animalia, you have vertebrates and invertebrates. I hope you'll know the difference vertebrates and invertebrates. What is vertebrates and invertebrates?
Organisms having backbone are called vertebrates.
Hmm.
Without backbone are called invertebrates.
Yes. Who said that?
Harish ma'am.
Harish. Okay. So this is mainly the classification about invertebrates and vertebrates. All of us know that. All of us know that. So this is. I hope you all will remember this. This is easy to remember. Three kingdom classification by Haeckel. This you
Yes,
will
ma'am.
remember. All of you can remember this. Then by Whitaker. Whitaker, I told you, American ecologist in the 19th century. He gave this, he proposed this in the 19th century. This is the five kingdom classification by Whitaker. Hmm? See, he has divided the kingdoms into Monera, Plantae, animals, Protista, and fungi.
Fungi.
Yeah. So here it is Monera, Plantae, animal, animalia, fungi, and protista. This is the five kingdom group. Under Monera, he's placed bacteria and cyanophyta. Under bacteria, he's placed bacteria and cyanophyta. Under Plantae are the group of plantaes which you have, we have discussed that Trichophyta, Bryophyta, hmm, Rhodophyta, Chlorophyta, Rhodophyta, all these come under, these are the different groups of plantaes. And fungi, all this. Osmocola, Cota, Ascomycota, Basidomycota, Zygomycota, all this. Mixomycota, Ascaromycota, all this come under fungi. Okay? And then under animalia or the whole big group of animals. All this comes under animalia. And then protista. Protista, he is placed under protista, he is placed what? Can you all read this? Under protista, he's placed the prokaryotes. Under protista, he's placed the prokaryotes, mostly unicellular. Okay. This is the differentiation table. This is the table of difference between the five groups. If you all want to note down, you note down, or if you all can remember. I don't think you can remember. Better to note it down. So he's differentiated the five groups between depending upon the cell type, the cell organization, cell wall, nutritional class, mode of action, nutrition, motility, differentiation of the tissue, and reproduction. Okay, depending upon all this, he has differentiated the five groups. So we'll do it one by one. Under Monera, he is placed, see over here, under Monera, what it was? Bacteria and Cyanophyta. Okay, so this cell type is prokaryotic cell type. We have studied the two cell types, no? Prokaryotic and eukaryotic. Yes or no?
Yes, ma'am.
Yes. So under this, under eukaryotic
Yes,
cell
ma'am.
type, yeah, under eukaryotic cell type, you have plantae, you have animals which have the eukaryotic cell, and you have the fungi which have the eukaryotic cell. Okay, and under prokaryotic cells, he has the protista group, the monera group. Monera group. What is the difference between the monera and the protista group? Let us see that. And a plant and animal, we know. We know the cell type of plant cell and the animal cell. These two we know. We'll see the difference between the monera and the protista, both cell types being prokaryotic. What is the difference? See, cell organization is unicellular. Cell wall is present in these cases, and cell wall is absent in these. When you say protista, cell wall is absent. Nutritional type, phototrophic. These are heterotrophic and phototrophic. What do you understand by heterotrophic?
They depend on other for food.
Other
They depend on
organisms for
other
food.
depend on other organisms for food.
Dead and decaying organisms, heterotrophs. Okay. So that was nutritional class. Then mode of nutrition, absorptive or ingestion. Here also absorptive or ingestion. They can be motile or non-motile. The bacteria, as we know, we have studied bacteria, they can have flagella and they can be motile. Flagella is responsible for motility. Okay. So some have the flagella, some do not have the flagella. So flagella, they can be motile or non-motile. Differentiation of the tissue, absent, may be present. Reproduction can be asexual and sexual. Over here also asexual and sexual. Okay. So here is mostly clarified depending upon the cell wall. Okay. All everything is same depending upon the cell wall is classified them into Monera and Protista. If cell wall is present, this is Monera, and we place them under Protista if cell wall is absent. Okay. So that is the two groups which he has done. And then coming to plantae and animals, this we know cell wall present and can be absent in few. Nutrition, phototrophic and heterotrophic, mode of nutrition, absorptive and ingestion. Okay, we understand the difference between absorptive and ingestion, no?
Yes, ma'am.
Yes. And motility, mostly non-motile, or plants are non-motile, and animals motile. Differentiation of tissue absent, differentiation is present. Reproduction can be asexual and sexual. Okay. Then coming to fungi. Cell type, eukaryotic cell organization, multicellular, hmm? and cell wall is present. Nutritional class, heterotrophic, mode of nutrition, absorptive, motility, non-motile, differentiation of tissue absent, reproduction, asexual and sexual. Okay, so this is how he is classified them into five major groups. That is Monera, plantae, animals, protista, and fungi. This is by whom? This classification is by whom?
RH
RH
Whittaker.
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 Yeah,
RH Whittaker.
yeah. All of you are understanding now.
Yes, ma'am.
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes, ma'am.
Classification topic itself is not very attractive as far as I guess for students. But if you all are understanding and if you all are able to take it and you remember it, it's good, because it is the whole unit is that this whole unit is you have classification by Calbus, Whittaker, Whittaker. You have to study that Haeckel and Whittaker, and then you have to study classification of viruses. Okay, did you study classification of virus in your 12th?
No, ma'am.
Yes, ma'am.
No, ma'am.
No, ma'am.
No,
No,
ma'am.
ma'am.
Who
No,
said
No.
yes?
ma'am.
No, ma'am.
I heard one yes. Who said that? One person said classification of viruses you have done. Who is that? Tell me your
Ma'am, Sri
name.
Akshara, ma'am.
Shri Akshara, you have studied classification of virus?
It's about prions, virus, viroids. Is that the thing, ma'am?
Yeah. What did you
Yes,
study?
ma'am, we did it.
Not the structure of virus. Structure of virus, I think you have done the TMV and HIV structure, no? All of you?
Yes,
Yes,
ma'am.
ma'am.
Yes, ma'am.
Yes,
Yeah,
ma'am.
I know. You all have this TMV and HIV structure in your syllabus, but I don't think classification of virus you have.
We
Yes, ma'am.
don't have, ma'am. We just
We don't
discussed
have,
about
ma'am.
acellular organisms.
Yeah, I know that because classification virus is you cannot have it at inter level. TMV and HIV structures you have that also in your unit. Okay, it's all easy because second unit almost 70% you have done in your inter, but little bit like classification of virus and some new topics will be there. Did you do Burgess manual?
No, ma'am.
No, ma'am.
No, ma'am.
No,
That
ma'am.
is one
No,
more.
ma'am.
Burgess manual is one more classification what you have in this unit. So this unit is majorly made up of all classifications. These three classification, then you have the Burgess manual classification, and then we will have to do the virus classification. Virus classification is big topic and important topic. You have general characters of virus, you have virus classification, then you have TMV and HIV structure. That you all know, but we have to do it again. Hmm? So this was about Whitaker. Is it clear?
Yes, ma'am.
Clear
Yes,
to all
ma'am.
of you?
Yes,
Okay,
ma'am.
Yes,
he
ma'am.
has divided them in. This was in the 19th century. He's divided them into five kingdoms. He's given this was these are the different classification proposed by different scientists. Okay, so we cannot say that this is correct, this one is correct, because that is the way they have proposed the classification. That is their point of view of classifying. Okay, so that is how they have done it. Whitaker has showed it in a five kingdom way, whereas your Calvus, next is Calvus. What you're doing?
6.
He has given a three kingdom classification. Okay,
Three
I'll show you that.
domains.
The next what we're doing is Calvus, which is a three kingdom classification. Okay, Calvus is again an American microbiologist. Okay, he is in the 19th century. This is the one which was the latest one which was given to us was by Calvus system of classification. American microbiologist. You can see this. He's divided into three. He's again proposed a three kingdom classification: bacteria, archaebacteria, and eukaryota. Hmm? Can you see this, all of you?
Yes,
Yes,
ma'am.
ma'am.
Yeah. So here he has classified the bacteria into different groups, and he's given the archaebacteria into different groups. Then he's put the eukaryotes separately over here. Okay, which includes the animalia and the plantae. So we'll see the bacteria which is classified into. Is classified green filamentous bacteria, green gram-positive bacteria, okay, and then spirochetes, then proteobacteria, then cyanobacteria, then plantomyces, then bacteroids, then thermotoga and aquifex. These are the different types he's classified bacteria into. Then he put archaebacteria in a separate group. Okay, archaebacteria is placed in a separate group, that is methanosarcina, methanobacteria, methanococcus, T-cellular, thermoproteus, and pyrodictum. You need to remember these words. Okay, you need to remember these names of different bacteria. And then the eukaryota. Okay, he's put it into eukaryota, a different group which has eukaryotic cell. Okay, he's put all the eukaryotic cells into a different group that he's classified as eukaryota, and then he's put into it the slime molds, the entamoeba, all those which have eukaryotic cell. Okay, animals, fungi, plants, ciliates, flagellates, trichomonads, microsporadia, and diplomonads. Hmm? Calvus, you have not done, you said, no?
Yes, ma'am.
We have done, but not in detail, ma'am.
Whitaker and Haeckel also you have done.
Heckel
Yes,
we
ma'am.
have not
Whitaker,
done, ma'am. But Whittaker we have
ma'am.
done.
Haeckel is an easy one which you can remember. Haeckel is just a three kingdom classification which you can remember. Hmm? Then Whitaker is a five kingdom one. I told you all that is also easy to remember. When you make it into tabular form, it becomes easy for you to remember. Anything, if you put it in a tabular form, it becomes easy for you to remember. Hmm? Over here, eukaryotes, you can see he's put into animals, fungi, plants, ciliates, flagellates, trichomonads, microsporadia, and diplomonads. All this which have the eukaryotic cell are put into this group. Okay. So this is a universal phylogenetic tree. This is called as a phylogenetic tree. Okay. You understand a tree, no? Generally, tree structures are used to show classifications or genetic traits. Okay, so this is a phylogenetic tree. If it shows you about the genus and the species and all, it is called as a phylogenetic tree. Have you heard this term phylogenetic?
Yes, ma'am.
Yes, ma'am.
Hmm. Okay. So when it gives you details about the species and the genus, if the tree is giving you details about species and genus, it's called as a phylogenetic tree. Okay. From comparative rRNA sequencing. Okay. Now this was in 1993. This was given in 1993. So this is the latest one you can see. Hmm? Can you recollect and tell me about Haeckel? What did we do? Haeckel, Haeckel divided into which groups? Who will tell me?
Protista, Plantae, Animalium.
Hmm.
Three
Protista,
kingdoms.
Plantae, and Animalia. Who said that?
Gautam, ma'am.
Gotham. All of you, do you remember? We just nodded Haeckel.
Yes, ma'am.
Yes, ma'am.
Yeah,
Ma'am.
this is the easiest of all.
Yes, ma'am.
Haeckel's is the easiest of all. I don't think there should be any problem in remembering the Haeckel classification. For short answers, they sometimes ask Haeckel also. Okay. So remember this: Protista, Plantae, Animalia. Easy to remember. Under Protista, you have all those prokaryotic cells with algae, fungi, and protozoa bacteria. Okay. And under Plantae, you have bryophytes, pteridophytes, gymnosperms, angiosperms. Under Animalia, you have invertebrates and vertebrates. You can remember this flowchart. Yes?
Yes, ma'am.
Yes. Hmm. Then Whitaker, who will tell me Whitaker? Whitaker classified into 5 groups.
Monera, Protista,
Monera,
Fungi,
Mono
Plantae, Animalia.
Monera protista
animalia.
animalia.
Yes. Girls don't answer my questions. Huh? Okay. He's classified into 5 kingdoms: Monera, Plantae, animals, Protista,
Protista and
and
fungi.
okay. And what are the differences? He's depending upon the cell type, cell organization, cell wall, nutrition class, mode of nutrition, motility, and reproduction. He's classified them into 5 kingdoms, that is Monera, plantae, animals, Protista, and fungi. Under Monera, it is prokaryotes, and under Protista, also prokaryotes. But
Prokaryotic.
the difference is here: Monera, cell wall is
Cell
present,
wall is present, cell wall is
cell
absent
wall
in
is
Protista.
absent. Okay. Note down if you all tend to forget. If you all think you will forget, please note down the differences. Hmm? Because it is important. I told you, classification question is compulsory in the paper. You get one question on classification, either long or short. You get it. Then under Plantae and Animalia, they have mostly eukaryotic cell. Eukaryotic cell mostly multi, and you know these are multicellular organisms. Yes, we all know what is multicellular, what is unicellular. What is multicellular? What is unicellular?
Organisms having single cell are called unicellular organisms.
Hmm. Multicellular?
Having more than one cell.
So that mostly is Plantae and Animalia, which are made up of many cells. They are made up of many cells. Okay, as is our body. As is our body. Okay, we are also a multicellular organisms. We our body is made up of many cells. You know that. Then what is it? Then what is the difference? Cell wall between plant and animals. Okay. Then their mode of nutrition, phototrophic and heterotrophic. We know these animals are heterotrophic. Then what remaining? Motility. You know, main difference is motility because these are non-motile, these are motile. Okay.
Motile.
This is about plantae and animals. Then protista. Protista, what does this? Same as they are prokaryotic, but only difference is cell wall is absent. Okay.
Absent.
Nutrition can be heterotrophic or phototrophic. Okay, we know what is heterotrophic, what is phototrophic. We have discussed just now. And then can be motile or non-motile, and reproduction can be sexual or asexual. Okay, so that is
Well,
protista. Then fungi. Fungi, what it is? It is eukaryotic. Cell type is eukaryotic. Present cell wall is present. Okay. Mode of nutrition is heterotrophic. They depend on others for their food. They depend on any dead and decaying matter for their food. Okay. Then absorptive, non-motile, and because they grow in one place, they are non-motile. Okay. Clear? This is Whittaker
yes, ma'am.
classification. Clear, all of you?
Yes,
Yes,
ma'am.
ma'am.
It's easy to remember, I guess. When you make the table and you learn it, it's easy to remember. You can
Okay,
also
ma'am.
draw the chart. This also you can draw. You have to remember the names, and then you can draw this also. It is done. If it comes, you have a long question. Children do this. They draw this also. Okay. The only thing is you have to remember these names group wise. Plantae, fungi, and these groups you need to remember by group name wise. That's it. You can draw this also. Then is Calvus. Calvus, I just now told you all. Who will tell me about Calvus?
Three system of classification:
bacteria,
bacteria,
archaea, to eukaryote.
We can do class.
archaea, eukaryota.
Eukaryote.
Bacteria,
Archaeeucaryota.
Archaea and eukaryote.
yeah. Under bacteria, what all is put?
Spirochetes, proteobacteria.
Hmm. Who is this telling me the answer?
Harish, ma'am.
Okay. What else? What
Gram,
about other?
gram positive.
Yeah. Under bacteria, what else is placed?
Green filamentous bacteria.
Yes. And
Acufex.
Xanthomyces.
mostly all the bacteria come under this. Okay. Mostly it's all the bacteria which come under this. See, cyanobacteria, proteobacteria, spirochetes, plantomyces.
Bacteriotes.
Yes. Okay. And Archaebacteria is placed in a separate group. Archaebacteria, just learn these names, okay? When you have to draw a flow chart, when calvus, if the question is calvus, you can easily do it. Hmm? You just have to learn these names. You have already told me a few names. You have to just learn a few more. Hmm? So this all comes under bacteria, and under Archaebacteria is placed the whole group of Archaebacteria. Methano means Methano means what? Methanobacter. You have studied this.
Methane producing.
Methane.
Yeah.
Methane producers.
Yes. And they survive in tough conditions where they produce methane gas. Hmm? So
Yes,
under
ma'am.
this
Gut of
comes
animals.
methanosarcemia, methanobacteria, methanococcus. Okay. Then
Cyanogen.
thermo
High
They
resistant
live in
temperature.
temperature.
Again,
Okay.
withstand high temperatures.
Yeah,
High
thermo
temp.
means high temperature. And under Archaebacteria, you have one more group, halobacteria. What does halo stand for?
It can
Salty
HAL.
withstand
areas.
in
Saline
salty
conditions.
areas.
Salty areas. Ma'am.
Who told me first?
Salty environment.
Gautam. Ma'am. Gautam.
Halo means salty. Okay, HALO
Salty
means
areas.
salty conditions where they can survive. That is under seabeds. Okay, these are found in seabeds mostly. So that group is that also comes under Archaebacteria. Halobacteria also
Archaeobacteria.
come under Archaebacteria. And halobacteria, they survive for many generations and decades because they are in the seabed and can survive in those conditions. Okay. So the Archaebacteria is the mostly those group of bacteria which are used to study the archaeological studies. Hmm? You are aware of the archaeological studies, no? Which we do for our different forts, and if we are doing any sculpture, if they want to know the decade when it was done, with the bacteria study the Archaebacteria present on that. Yes or no?
Yes, ma'am.
Are you all aware of that?
Yes, ma'am.
There are various archaeological studies going on. You know what is archaeological studies, no?
Yes, ma'am.
All of you know. You know what is archaeological study. Archaeological study is when they're studying about the different forts or our age old sculptures. If they're studying, they're doing any study on the age old temples. They are studying. That is the archaeological study which they do. Archaebacteria present over there. They study that. So Archaebacteria is able to survive for decades over those sculptures and temples and forts, rocks. Okay, on the rocks it survives under tough conditions where there is no food, no water. These are the bacteria which survive Archaebacteria. Okay. Then coming to Eukaryota, where he is classified into animals, fungi, plants, ciliates, flagellates,
Flagellates.
trichomonads,
Monads.
micropodia, diplomonads. Okay, all this come under eukaryotes. So Calvus has classified the bacteria micro world into three
Three
kingdoms:
kingdoms.
bacteria, archaea, and eukaryotes.
Eukaryota.
Under bacteria is placed all the
Yes.
bacteria. Archaebacteria is placed separately. Under eubacteria, eukaryota is placed all those which have eukaryotic cell. Hmm? So all these have the eukaryotic cell. So he's placed them separately. Okay. So this is a three kingdom classification given by Calvus in 1993. Hmm? I hope you all will remember this.
Yes, ma'am.
Is it clear?
Yes, ma'am.
Yes, ma'am.
For all of you, I hope Calvus, Whittaker, and Haeckel classification is clear.
Yes, ma'am.
Yes,
Hmm?
ma'am.
Yes, ma'am.
Yes, ma'am.
You should be able to remember it and also write it in the notebook. Okay. So, shall I conclude? We have one more big classification to be done after this, that is the Bergey's Manual of Classification. I don't think we can do it now. It is a big Bergey's classification is a big one, but this I think you all are clear with. If you all want to take the images, you can take so that you remember. I know you cannot note it down, but if you want to keep the images clicked so that you remember and you recollect, you can click it. Anybody wants to click it or shall I close it?
Already taken, ma'am.
Taken. Or Vitaka, do you want to take this?
Already
Yes,
taken, ma'am.
ma'am.
Taken,
Taken?
ma'am.
Taken, okay.
Taken, ma'am.
Taken, ma'am.
Okay. And I hope for Haeckel also you all will remember.
Yes,
Yes, ma'am.
ma'am.
Yes, ma'am.
Okay. Okay. So today's class, did you all understand everything?
Yes, ma'am.
Yes,
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 ma'am.
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 Is
Yes,
everything clear?
ma'am.
Whatever was taken to you all today?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
All what have we taken? We have taken the structures, we have taken classification, then we have done gram positive, gram negative, cell wall, cell structures. We have done today difference between gram positive and gram negative cell wall. You all remember?
Yes,
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 ma'am.
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 Yes, ma'am.
Or it was too much of overloading for you all?
No, ma'am.
Is it okay?
Yes,
No,
ma'am.
Okay,
ma'am.
ma'am.
Yes, it is.
I repeat it many times and I go slow so that it doesn't become too much for you all, because you will have just passed from your inter. You all were used to 40 minutes of class or 1 hour of class?
1 hour.
1 hour, ma'am.
1 hour,
Yes,
ma'am.
ma'am.
Your wish, madam.
You can switch on your videos. Switch on your videos, whoever can. So you all were used to 1 hour of your class.
Yes, ma'am.
Yes, ma'am.
In Narayana and all also they take 1 hour only, or they take 2 hours?
1 hour,
1
ma'am.
2
hour.
hours, ma'am.
1 hour. 2 hours. Who said 2 hours? Deepak said 2 hours, I think.
Gautam.
Gautam said 2 hours. So that's what, 2 hours of class. I don't know how much you all can take, but I go slow and I repeat it many times so that you all understand and it's not very fast for you all. Hmm? At any point you all want me to repeat, you can tell, I will repeat. Okay?
Okay,
Okay,
ma'am.
ma'am.
Okay,
Okay.
ma'am.
And Akhila is not switched on.
Ma'am, there is some camera issue, ma'am.
Camera issue. Okay. If everything is okay, did you understand everything?
Yes, ma'am.
Okay. Kamari, huh? Manohar Kamari.
Yes, ma'am. Navyashri.
This is Navyashri, huh?
Yes, ma'am.
Okay. Navyashri, did you understand everything?
Yes, ma'am.
Okay. Hafsa, कुछ problem है तो you contact me again.
No, ma'am, but it is important to wear hijab. But I can't.
Pardon, I didn't get it.
Mom, it is important to wear hijab, but now I can't.
Okay. 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 Thank you, all of you. See you in the next class, I think next week.
Thank you, ma'am.
Thank you,
So,
ma'am.
see you all in 2020,
Thank you, ma'am.
Thank you, ma'am.
2021.
Thank you, ma'am.
Thank you,
And
ma'am.
happy
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Okay. Bye-bye. See
Bye.
you all
Bye,
in
ma'am.
Bye, ma'am.
2021.